Okay, this final question is an absolute gift of a question, 7 marks on describing transcription and translation. People made a really good effort at it. I think the one thing I would just point out is this pencil means that there is a QWC, quality of written communication mark available. So if you got all the marks, that means that you were awarded that mark as well, but I'll just go through how that's done. So for this question, I think the mark scheme is absolutely fine to just have a look at. So you've got marking points 1, 2, 3, 4, 5 for transcription, then 5 for translation. If you are talking about how a nucleotide base sequence makes a polypeptide, you need to mention both of those. So you've got some guidance there. So you've got 5 marks for this and 5 marks for this. Oh, sorry, 5. So transcription, not many people mention point number 1, that just DNA is transcribed into mRNA, and then tell me how it's done. This second point came up a lot. Just remember that free activated nucleotides. Complementary base pairing was done really well using a template strand, and that is catalyzed by RNA polymerase. That was generally done really well. And then on translation, the mRNA moving to the ribosome was good. tRNA molecules bind to mRNA. Okay, the anticodons. Remember that tRNA has this cloverleaf structure, and on this top bit you have the anticodon, and on the bottom bit that tRNA has a specific antibody. Okay, and that's amino acid, and that specific amino acid is related to the anticodon. So tRNA molecules will bind to mRNA in the ribosome. The anticodons will match to the codons and bring a specific or correct amino acid because they're attached to tRNA. The final bit wasn't done very well. So remember, in the ribosome, we might have 2 of these next to each other, and we have 2 amino acids next to each other, and the ribosome will bond those together to form a peptide bond. Again, that one will be reinforced when we do protein structure, so don't worry if you missed that one, but it's a nice point if you could get it. Now, you could get 6 marks for that content, and then down here, award QWC if there are 2 marking points from 1 to 5, and those have been awarded before 2 marking points from 6 and 10. So that really just guards against candidates who are just sort of fireballing out sort of random bits of transcription translation, hoping that they hit some marking points. If you are being very logical about this happens and this happens and this happens with these processes, using bullet points can really help you there to structure those answers. Then you can pick up that mark very, very quickly. So even if you got full marks on that question, just go back, see if you missed out any of those marking points. I'm not sure anyone got 10 out of 10, but lots of people got enough to get 7 out of 7, so that's great. But yeah, just go back, have a quick look and add in another colour, because the best way to do this, to do your feedback, is to use that different colour, because then you have a ready-made piece of work telling you under pressure, what do you forget? It's the stuff in the other colour. Okay, so well done. Like I said, I'm really pleased with how you all did in this test, really pleased to see you revising and working so well. And if you've got any questions, do just email me or ask me in the next lesson. Bye.